Okay, so let's get back to glycolysis. Remember, we're talking about three main pathways. If we're going to do this process of respiration, what we're talking about is taking glucose all the way down to carbon dioxide and water and making a bunch of ATP, as much as we can. So what are the three main pathways involved that we're going to talk about? What's the first step? Glycolysis. Basically, we're just going to chop the 6 carbon into 3 carbon units. And what do we get for doing that? We get 2 ATPs and some pyruvate. What's the next step if we're going to keep going? What's the main product that's valuable in the Krebs cycle? Do we make any ATP in the Krebs cycle?
No. I think something else, but I forgot.
We get a little bit of ATP. What else?
Carbon dioxide?
Yes, that's a waste product. That's where all of the carbon dioxide is that you exhale. All of your carbon dioxide waste product comes from the Krebs cycle. And we're going to talk about this mystery thing. Okay, so if we've gone through the Krebs cycle, then we go where?
Electron transport chain.
Electron transport chain. So we're going to pass electrons down a chain, and we're going to use the energy from that to pump protons. The protons are to come back through a turbine. Where are we getting the electrons?
Stripping them from something else.
Oxygen collects them at the bottom of the electron transport chain.
Stripping them from something.
We are taking them from breaking bonds, and those high energy electrons are being collected on electron acceptors throughout the process of both glycolysis and the Krebs cycle. Anybody remember the name of the electron acceptors, there's 2 of them, that are involved?
FAD.
So we do make a little bit of NADH in glycolysis. We make the bulk of it and some FADH2 in the Krebs cycle, and that's where we get our electrons for the electron transport chain. So let us walk through this process. Okay, so the first step, glycolysis, we chop glucose in half, we make 2 pyruvates, we generate some ATP, we also generate some NADH. And there's 2 main, we can sort of divide the reactions in glycolysis, which there are many. We can divide them into 2 parts, the investment part and the payoff part, because initially we actually have to put some energy into glycolysis. We actually use 2 ATPs to put high energy phosphates onto glucose. And that is actually in order to help us split the glucose. Splitting glucose is not simple. We have to put some energy into doing that. Once we do that, we generate 2 molecules which each have a high energy phosphate on them. And then in the next few steps, we take those phosphates back off and make ATP, and we also get a little bit of energy from splitting more bonds and make a couple more ATPs. So we use 2 ATP, we make 4 ATP, and the total net of glycolysis from 1 molecule of glucose is 4 minus 2. We can all do that simple math. Okay. Now we also make 2 NADHs in the process of doing glycolysis. And that is because when we are splitting these bonds, we're liberating high energy electrons, and NAD plus is the electron acceptor. It's electron carrier that we're going to use to collect those. So I'm simplifying this multi-step pathway considerably by saying that at some point we're taking 2 NAD pluses, collecting some high energy electrons. When we do that, it creates this. Now, remember, this is a redox reaction, right? So those high energy electrons are being taken from something. The thing we took them off of is oxidized or reduced. It's oxidized. We stole the electrons. That's the victim, right? That's oxidized. But we put them onto NAD+, creating NADH. So which form, NAD+ or NADH, is the reduced form? Which one has the electrons? This is the reduced form, okay? So we reduce some NADH. So reduced things have more energy by definition. They've got these high energy electrons, they now have more energy. We have this reduced form of NADH. We're going to use it later on, potentially. But for now, we just know that we made some ATP, we made some NADHs. So what we get from splitting glucose in half is some pyruvates, some ATP, and some NADH. Now, if we don't do glycolysis, we can do a couple of other things that are described in more detail in your textbook. So I'm just going to mention them now. The pentose phosphate pathway and the Entner-Duderoff pathway. And there's a couple of differences between these. So these pathways are used for a variety of reasons. They allow you to be more flexible. The pentose phosphate pathway is helpful because it means that you can metabolize sugars that aren't glucose, right? Things that have 5 carbons. So you can use a 5-carbon sugar instead of a 6-carbon sugar. And instead of generating NADH, you generate NADPH. And instead of generating 2 ATPs, you generate 1. And what you're doing actually is you take that 5-carbon sugar and you turn it into glucose. And then it goes through the process. But because you had this extra step, you don't get as much energy out at the end. Now, it's also useful because it's reversible, which means you can take glucose and make pentoses. Why would we want to do that? Where in the biological macromolecules do we have 5-carbon sugars that we need to make? DNA and RNA, right? Because those nucleotides have a phosphate group, a 5-carbon sugar, and a base. So we need to get that from someplace. We can also use this other pathway. The Entner-Duderoff pathway does not go through glycolysis. And only some soil, and these are actually nitrogen-fixing bacteria that tend to be living on the roots of plants. Some of these genuses contain species that do this process. And all you need to know is that it's another way to make energy and does not involve going through glucose. Okay, so basically what we're doing with glucose and glycolysis is we're oxidizing it. We're partially oxidizing it, and if we continue to do respiration, we'll go all the way down. If we stop here, then that's considered fermentation. In the glycolysis step, we're generating NADH and ATP. If we keep going, we're going to get more NADHs, FADH2s, and more ATPs. I just want to mention that glucose has a variety of possible destinies. So you can burn it all the way down to the ground, right? If you have the enzymes to do that. Why would you maybe not do that? Why would you sort of stop halfway through and do something else with it? So one of the reasons that we metabolize glucose is to get energy, in which case we're going to just go through the whole process of glycolysis, the Krebs cycle, electron transport chain, getting carbon dioxide and water at the end. But you could stop before that's done. You could stop actually anywhere. You could stop at the pyruvate step, or you can get sort of halfway through the Krebs cycle. What is in the Krebs cycle, what's going around the Krebs cycle, are actually intermediates that you can use to make amino acids. So if you need amino acids, one way you can do that is by stopping sort of halfway through the respiration process and siphoning off those intermediates to make something else that you need. So that's just to sort of try and emphasize that it's a very complicated web of metabolic reactions. For now, I want to keep going on this sort of simple idea of getting as much energy as we can. So once we've done glycolysis, where do we go? If we're going to get all the energy that we possibly can, we're going to go to the Krebs cycle. So how do we prepare for that? We've got this 3-carbon pyruvic acid, pyruvate, and we oxidize it, so we steal some electrons. We decarboxylate it, so we take off one of the carbons, and that turns it into this thing called acetyl-CoA because it's joined with a coenzyme. So now it's a 2-carbon molecule. So what we do with our 3-carbon pyruvic acid is we strip off one of the carbons, making carbon dioxide, and we also generate another molecule of NADH because in this process we're oxidizing it, meaning what? Pyruvate, what's happening to pyruvate if it's being oxidized? The definition of oxidation, what is it? They're stealing electrons, okay? So where are those electrons going? They're going on to NAD+, we're making NADH, which is the reduced form. And so now we have more high energy electrons stored on this molecule. The whole point of doing that is to make acetyl-CoA so we can go into the Krebs cycle. So here we go. Now we're at the Krebs cycle. So our acetyl-CoA joins up with this oxaloacetic acid forming citric acid, which is the reason that this cycle is sometimes called the citric acid cycle. So we take something that's got 2 carbons, we plop it on something that's got 4 carbons, we make citric acid that has 6 carbons, and then we go around the cycle doing little modifications to that molecule. Occasionally we steal some electrons. So what's happening here? We go from isocitric acid to ketoglutaric acid. We are oxidizing this molecule, right? So this is the oxidized form. We're reducing NAD to NADH. We're collecting some high energy electrons. We're stealing more energy. Also, we're ripping off one of the carbons, making carbon dioxide. And those 2 processes happen at the same time because when we rip off the carbon, we're actually liberating the energy in that bond, which we collect on NAD. So we keep going around the cycle, and at a certain point we make a molecule of ATP. So given that there's no hydrogen ions being pumped anywhere and there's no electron transport chain, what form of ATP generation is this? We had 3 kinds, right? Substrate level, oxidative, and photophosphorylation. There's no sunlight in this process. There's no electron transport chain. So what is this? This is substrate level phosphorylation. So we make a little ATP, not very much. Okay, we keep going. We ripped off one of the carbons. We rip off another carbon. Now we've got a 4-carbon molecule, which just keeps on going. And every once in a while, we do a little rearranging and we steal some more electrons, right? So oxidize this succinic acid to make fumaric acid and collect the electrons on this. Very similar to NAD, it's called FAD. Keep going, we rip off some more electrons, collect them on NAD. So at the end of the Krebs cycle, what have we made? What are sort of the products? We took this 2-carbon molecule, we plopped it on a 4-carbon guy. We ran around the cycle, we stripped off some of the carbons, now we have 4 carbons again, ready to accept 2 carbons from the end of glycolysis to go around and more carbons and take them off and get more carbons. What are the primary products of this cycle? What are we getting out of it? A lot of NADHs, a little bit of FADH2s. And are we making any ATPs? Yes, we are, right? And the waste product, carbon dioxide. Okay, so all of those electrons that we ripped off, those high energy electrons, we stored them on NADH and FADH2, the reduced form of these electron carriers. Where are they going to go and drop their electrons? What's the next step? Say again? The electron transport chain. Okay, so here we go. We get these electron carriers, these NADH and FADH2s that we got them from glycolysis a little bit and mostly from the Krebs cycle, and we deliver them to a series of carrier molecules embedded in a membrane, and then we pass the electrons off to the first guy in the chain, and he passes it off to his friend next door, and so on until the tired electrons are collected by the terminal electron acceptor, which in many cases is what? Usually terminal electron acceptor at the end of respiration, oxygen, unless it's anaerobic respiration, in which case it's something else. So as the electrons are transferred, each transfer is from something that is less electronegative to the next one, which is more electronegative, so that process is easy and it releases some energy. So it's like the electrons can relax a little. They're going to someone they like even better, or that likes them a little better. So they relax, they give up some energy. They relax again, they give up some energy. That energy is used to do what? pump protons. The machines that are accepting those electrons also happen to be proton pumps. So protons get pumped across the membrane. Why, if you want to take a proton from one side of a membrane to the other, why do you need energy to do that? Part of the reason is because you're going against the gradient, but you would need energy regardless, unless you're going through a pore. Okay, if we're not going through a pore, we have to use energy to do that, partly because protons can't easily cross membranes because they're charged. Even though they're tiny, just one proton is so little, but they're supercharged, and the hydrophobic tails inside the lipid membrane doesn't like that. Okay, so the electron transport chain, you've probably heard this before, produce ATP by chemiosmosis. What that means is that as we're pumping those protons, yes, we're pushing them to the other side of the membrane where there's already lots of them. So we're using that energy to create a gradient. Those hydrogen ions would like to come back. They can't, though, because the membrane's in the way. What are they coming back through? They're allowed to come back through this very specific channel. Say again? No, although that is a good guess because, yes, the membrane is semi-permeable. It's just not, unfortunately, permeable to protons. They're locked out. What are they coming back through? When they come back, they're coming back through something. Now, I want you to think about it this way. It's as if we're pumping water to the top of a dam. Okay, now the water's all up there. And we're letting it come back, but when you let water come back through a dam to generate energy, you don't just let the water come in anywhere. You only let it come through a special channel. That channel is connected to a turbine. So the energy of the water falling drives the turbine around. That kinetic energy is used to make electricity, right? In a very similar way, we're going to let those protons fall down through a channel, which happens to be called ATP synthase. And it is actually a turbine. It will spin, and in spinning, it makes ATP. So we have to talk about the components of the electron transport chain. They're large protein complexes, so that means that each of the electron acceptors in the chain is actually a multi-protein complex. And as we've already discussed, many of them, not all of them, are proton pumps. There are three types: quinones, cytochromes, and flavoproteins. I'm not going to ask you to memorize a lot of detail about these. I just want you to sort of be familiar with this terminology, especially because you'll hear this a lot, cytochrome, and we're going to do tests for cytochrome C oxidase. So cytochrome C oxidase is one of the electron acceptors in the transport chain. So here's just a sort of a nice cartoon of this process. So there's the membrane. All of these guys are embedded in the membrane. Let me ask you this question. Have you ever wondered why the electron transport chain is in the membrane? Why is it not just like floating around in the cytoplasm the way that glycolytic enzymes are, or the Krebs cycle? Why are they in a membrane? It's partly, that's a really good point. It is partly that you have to establish a gradient. You can't do that unless there's some sort of partition from one side to the other. There's another reason. Electrons are being handed from one to the next. That means that they have these guys that are taking the electrons and handing them off, they have to be really close together and super organized in a particular way. And you're not going to have that so easily if they're floating around in the cytoplasm, right? If you're in a membrane, it's a lot easier to organize them. So yes, it is precisely those two things. It is that you need to organize them, and also you need to establish a gradient. So here is our complex 1, complex 2, 3, and 4, and then at the end, here's the channel through which we're going to have our protons come back. So as the NADHs and the FADH2 come, they drop their cargo off. So NADH here is the reduced form. It drops its electrons off with this first complex and turns back into this NAD+, which is the which form, oxidized or reduced? This is the reduced form, so what's that? Oxidized, right? Because this guy basically stole the electrons. Now, this guy who's been oxidized, where's he going to go? What do we do with him? We throw him away? No, where does he go? All the way back to glycolysis or the Krebs cycle. He's going to be used again. He's recycled. F-ADH2 does something similar. It actually drops its electrons later in the electron transport chain, which is why you don't get as many ATPs out of doing that. When we do the math, you'll see why that is. And as the electrons are dropped off and passed down, these machines are pumping protons from one side to the other. And on this side, the proton concentration is really high, which means what about the pH? It is acid, it's low, right? And on this side, it's going to be neutral. So now we have a whole bunch of hydrogen ions up here. And what do they want to do? There's lots of positive charges, they're crowded, they don't like each other very much. There's a lot of space, it's so nice over here. So where are they going to go? They want to come back, they can't cross the membrane, but they can come back through this channel, which just so happens to be just like the turbine in a dam. It is a turbine, and as the hydrogen protons come back through here, it spins the turbine. That kinetic energy is used to make ATP from ADP and inorganic phosphate. Now, another interesting thing that happens is as the electrons are passed, they are dropped off on oxygen, usually, right? If it's aerobic respiration, they're dropped off on oxygen at the end. That creates a negatively charged oxygen, which, by the way, is dangerous. We don't want that. And also, that is going to attract what? It's a negative charge, so it's going to attract what kind of things? Positive charge, like protons that happen to be floating around. So this negatively charged oxygen grabs whatever protons are around, which means that we're driving protons across, and any ones that are left behind we're going to use up making water at the end, right? So we're really, in both ways, establishing this gradient. And the primary product of that is going to be water. Okay, so that's the whole process is called chemiosmosis, and about 3 protons are used to make a single ATP. I'm going to stop the lecture here, and we're going to finish it up next time. We're going to talk about the difference between this process of aerobic respiration and the process of anaerobic respiration, which you already know, which is what? What's the difference between aerobic respiration and anaerobic? It's the terminal electron acceptor. It's just a slightly different molecule. And we're going to talk about the difference between that and fermentation so that you're really clear on the differences. So the difference is huge, but most people get fermentation and anaerobic respiration mixed up because both of them can happen without oxygen, but they're very different. And different organisms use a variety of strategies. Some of them can do aerobic respiration, anaerobic respiration, and fermentation. Some of them can only do one of those things. Some can do 2. And we're going to talk about those distinctions in the next class.